Okay, rest of the time, we'll introduce a little bit on the variation. We learn genetics, now we want to learn more on the variation. What's the meaning of variation? Change, right? So variation means change. Well, related to biology or microbiology, when we talk about variation, we are talking about mutation or other type of change. So we'll learn about gene mutation and also gene transfer and recombination in bacteria. And we'll learn transformation, conjugation, and transduction. Okay, the first one, gene mutation. Gene mutation can happen in different ways, in a lot of ways. For example, we have the DNA in our cell, and they are relatively stable. So we want to keep them stable. You don't want them to change all the time, but the DNA is not as firm as we thought. They are always changing. And for example, there might be like base substitution. Like you have the bases ATCG, so maybe one base will change to another type. Or deletion, part of the gene, part of the DNA gets deleted. Or insertion, like a DNA fragment inserted into the chromosome. It's possible, and it happens all the time. And also the DNA duplication. At the beginning, you might have only one copy of the gene, but for some reason, that gene makes another copy at another location. It's possible. Or the DNA inversion. So the direction of the gene change. And these mutations can arise in different ways. It can arise spontaneously. So without any induction, when the bacteria replicate from one generation to another generation, there might be some mistake happen in the DNA sequence. Even though the frequency is very low, it might be 10 to minus 6 or 10 to minus 8, but it happens slowly with low frequency. But after a long time of cultivation, there might accumulate more and more mutation. Or mutation can be induced by either physical factors or chemical factors. Physical factors include like UV light as a mutagen. We learned that last time that UV light can form the TT dimer in DNA structure. And that TT dimer is a scar in the DNA sequence. So the bacterial cell would cut out the scar and repair it. But when the DNA is repaired, probably some mistake can happen. So the basis might change. So this is a physical mutagen. Or some chemicals can induce the DNA sequence to change. So these are the mutations in bacteria. It can happen in a lot of ways. And well, from the side of evolution, those mutations are not totally bad. Okay, some mutation might be lethal. The cell will die. Well, that's bad for this specific cell. But sometimes the mutation is neutral. It does not kill the bacteria immediately, but it contributes the diversity of the bacteria, so that the DNA sequence will become diversity. When the environment changes, the bacteria will be able to adapt to different type of environment. Okay, when the environment change, like to dry condition, probably a small portion of the bacteria can adapt to this dry environment. They can survive. So mutation is not totally a bad thing. Sometimes it's good for the whole population. Okay, and we'll focus on gene transfer that's the most important part. Gene transfer is, well, the DNA gene can transfer from one species to another species, from one cell to another cell. So there are several general properties of gene transfer in bacteria. The first one is unidirectional. We know this word uni means one. So unidirectional means one direction. So this can only happen from the donor cell to the recipient cell. So the one direction. And usually the donor cell does not give the entire chromosome to the recipient. It can give maybe simply a plasmid, a part of its chromosome, not its whole chromosome. And gene transfer can also occur between species. It's not limited to any specific species. E. coli to E. coli is possible, but E. coli can also transfer their gene to Salmonella. That's also possible. Or even to other organisms, like bacteria can transfer their gene to plants. So that's a common method we use in the lab working on plants. They use bacteria to multiply the plasmid. Then they mix the bacteria with the plant so that the bacteria can inject or transfer the plasmid into plant cells. So it's a useful tool so that we use this method, you can, well, modify the genome of plant or let the plant cells express certain gene to produce something. So that's the general features of gene transfer in bacteria. Okay, the first type of gene transfer we are learning here is the transformation. Transformation is the gene transfer resulting from the uptake of a naked DNA from the environment. So, okay, a recipient cell take up a naked DNA from the environment. This naked DNA probably is released by other bacteria or other cells into the environment. Then the recipient cell will take up the DNA and integrate the DNA into its genome and express the DNA so that this recipient cell would gain another property. There are several factors affecting transformation. First, the DNA size and state. If the DNA is too big, it's not easy to get into the recipient cell, right? So you want the DNA in the proper size and also proper state. Because if the DNA is in open state, linear, long, loose state, they are very sensitive to nucleus, like DNAs. DNAs would cut this DNA into small pieces. So if the DNA is supercoiled, condensed form, it's easier for transformation. And they are more resistant to nucleus. And also, the competence of the recipient cell. What's the meaning of competence? Competence is the ability of the recipient cell to take up DNA. And interestingly, some bacteria, they are naturally competent. So which means that the bacteria are always ready to take up extracellular DNA. So we call them spontaneous competence, okay? Such as Bacillus, Haemophilus, Neisseria, and Streptococcus. The famous model we learned in high school is probably you know that story, the transformation of Streptococcus pneumonia bacteria. So they have two different type of colonies, smooth colony and rough colony. So if you mix bacteria from the rough colony with dead bacteria from the smooth colony, some bacteria will become smooth. You know that? So that's, well, actually natural transformation. It's because Streptococcus pneumonia can take up DNA spontaneously so that they will gain a new phenotype. And also, we need to think about the competence factors, environmental factors. And in the lab, sometimes if the bacteria is not spontaneously competent, we need to induce them. For example, E. coli, the most widely used bacteria in the lab, it's not spontaneously competent. We need to treat the bacteria with chemicals such as calcium chloride, so that after treating with calcium chloride, E. coli is able to take up extracellular DNA. Okay, so this is transformation. Let me show you mechanism of transformation. There are two steps. The first step is that, well, extracellular DNA needs to get into the cell, okay? This is a recipient cell. These fragments need to get into the recipient cell. But solely getting into the recipient cell cannot guarantee the transformation, because inside the recipient cell, there are a lot of DNAs. The DNAs would degrade the free DNA fragments. And only if there is homologous sequence on this fragment with a chromosome, so that after recombination, part of the DNA fragment get integrated into the chromosome, into the genome, okay? So that this fragment is stable. The genes carried on this fragment can be expressed so that this recipient cell will gain some new property. So this is two steps: uptake, then recombination. This is important, two steps. And yeah, sometimes the uptake, the DNA uptake by the recipient cell is not fragments, but it's a plasmid. That's also okay, because inside the cell, the plasmid can replicate by itself, and it's stable, okay? So, but plasmid transformation, yeah, it's widely used, but it's not, sometimes it's not considered a real transformation, okay? And this transformation technique is important for research and also for like clinical work. And in our research lab, we always use recombinant DNA technique to express some gene, to construct a plasmid, and study the function of the gene. Or in industry, you can make recombinant DNA and express some gene to produce something. This is transformation. Let me show you a short video on transformation. Hopefully it will work. Never mind. It's too long. Okay. Okay, the next gene transfer is conjugation. We actually learned this a little bit before. When we talk about the bacterial structure, we learned that a special structure called sex pili or F pili, and that pili can mediate gene transfer. Okay, this is the donor cell. It carries a F factor, which can express the sex pillars. Okay, this is a recipient cell which does not carry the F factor. Okay, the sex pillars here, the conjugation pillars, can grab the recipient cell, then retract to bring the two cells together. Once the two cells get together, there will be a channel formed between the two cells. And through this channel, the donor cell can transfer its DNA into the recipient cell. Okay? the single-strand plasmid is transferred into the recipient cell. And finally, both cells will carry the same plasmid, the F factor. So this is conjugation is the gene transfer mediated by the sex pillars. Okay? A special transformation with a special conjugation in bacteria is through the high frequency recombinant. This is a phenomenon is due to the integration of the F factor into the host DNA. So we don't know the exact reason, but sometimes this plasmid can be integrated into the bacterial DNA, the bacterial genome. And in this genome, the genes of the F factor can be expressed. So these bacteria also carry the sex pillars. And these sex pillars can mediate the gene transfer. Okay? Bring the recipient cell together with the donor cell, then the channel form, the DNA can be transferred into the recipient cell. But here, there's no plasmid anymore. The DNA transferred into the recipient is the genomic DNA, not the plasmid DNA. The long genomic DNA fragment into the recipient cell, then a recombination can happen so that the genome is modified. And this is the high frequency recombinant phenomena. We'll have a break and come back later. Okay? So, yeah, we are talking about the conjugation. A special type of conjugation is through the high frequency recombinant strand. So that's HFR. That's the bacteria strains with integrated F factor, okay, like this one, which can transfer their chromosome DNA, chromosome gene to the recipient cell with high frequency. So that's why we name this strain the high frequency recombinant strain. And it's a useful tool in the lab for research. Okay. The last type of gene transfer we are going to introduce is called transduction. So this is included in our textbook. Transduction is a gene transfer mediated or shuttled by bacteriophages. We learned bacteriophages just now. And, okay, so the gene transfer from donor cell to recipient cell with a bacteriophage as a shuttle or a carrier. There are two different types of transduction. One is called generalized transduction. In this generalized transduction, any gene can be transferred. So that's random, the genes transferred is random. Another type is called specialized transduction. Specialized transduction means that it can only transfer some particular genes. Usually, that's the gene adjacent to the integration site in the genome. You know, when the prophage integrate into the genome, so that's the integration site. Next to the integration site, some genes can be transferred. And the difference between generalized transduction and specialized transduction is due to the different mechanism. Let me show you how it happens. For the generalized transduction, it's mediated by the lytic phage. Okay? So this is what happened. The bacteriophage okay, the bacteriophage infect the host cell. Okay, once they get into the host cell, the donor cell, the bacteriophage replicates in the cell. So after biosynthesis and assemble, new viral phages will mature, then it will lyse the host cell and release. And when it releases, pay attention to the new viral particle here. You can see that in these new viral particles, there are some mistake that in this particle and this particle, it carries not the viral genome, but part of the donor DNA. So there is a mistake that the viral particle carry the donor DNA rather than its own genome. Once this viral particle is released, it's able to initiate another infection, to inject this DNA fragment into another cell, the recipient cell, but it cannot generate new viral particle. Once this DNA fragment gets into the recipient cell, if recombination happens, the DNA is integrated into the recipient cell genome. So that's the gene transfer from the donor cell to the recipient cell mediated by the bacteriophage. And we call this generalized transduction because this mistake happens randomly. So basically, any fragment, any DNA fragment can be packed into the phage particle by mistake. So there is no specialized, not particular gene packed or transferred. So this is the first type, the generalized. While the second type is called specialized transduction. This transduction is mediated by the moderate phage or the lysogenic phage. Okay? Only a certain donor gene can be transferred to the recipient cell. And it's mediated by lysogenic or temperate phage. And the genes transferred depend on where the prophage has inserted into the chromosome. And let me show you this in a picture. So this is a specialized transduction. This one, the bacterial chromosome, carries the prophage. Here, the prophage. It's already integrated into the bacterial genome. Okay, once induction happens, for whatever reason, there is induction, so that the phage genome is kicked out of the bacterial genome. Okay, so it leaves the bacterial genome to form the phage genome. But here is what happens. When the phage genome is kicked out of the bacterial genome, there is some mistake happen. The phage carries part of the bacterial DNA, okay? And this DNA is adjacent to the insertion site, okay? This phage genome will guide the biosynthesis and form new viral particles. Okay, when these new viral particles is released, it can infect another donor cell. That's the recipient cell. When it infects a recipient cell, it forms the prophage again by inserting its genome into the bacterial chromosome. Okay, now you can see that this DNA is carried from the donor cell to the recipient cell. And this fragment is not randomly picked. It's the specialized DNA sequences. Some DNA fragment adjacent to the site of insertion. Okay, So that is why we call this the specialized transduction. Okay, to summarize this part, I'd like to play this video for you.
Welcome back. In this video, we'll be discussing the bacterial genetics, and we'll talk about transformation, transposition, transduction, and conjugation. We'll learn their meanings and how to differentiate between them. So first, let's talk about transposition. The bacteria has its DNA in two parts, either the bacterial chromosome or the bacterial plasmid. The bacterial chromosome is much larger and more complex and contains most of the bacterial DNA. And the bacterial plasmid is a small part of the bacterial DNA that has left the chromosome and now is floating inside the bacteria. So transposition refers to the process of extracting a small part of the bacterial chromosome to become plasmid or reintegrating the plasmid into the bacterial chromosome. Keep in mind that if the bacteria want to share its DNA with another bacteria, it can only share its plasmid, not the chromosomes. So the plasmid is smaller and readily accessible. Just remember, transposition has to do with plasmid. Now let's talk about conjugation. So some bacteria have a sex balance, and we call these bacteria F positive, and other bacteria don't have this sex balance, and we call this F negative. Keep in mind that the sex balance is encoded by the bacterial plasmid, not the bacterial chromosome. So the F positive bacteria will link with the F negative bacteria and form the mating bridge. Then the F positive bacteria will make a copy of its plasmid, which encodes for the bacterial sex balance, and will transfer this copy to the other bacteria. So now both bacteria have a plasmid which encodes for a sex balance, and both are called F positive. This is the process of conjugation. Transformation, which is also known as competence, is the ability of the bacteria to take naked genomes or naked DNA from the environment and use this DNA. So for example, if we have some medium with some bacteria in it and we put some raw naked DNA which encodes for antibiotic resistance into this medium as well, this bacteria will be able to take this DNA from the environment and use it, and it will become antibiotic resistance. However, if we add a DNA lysing agent into the environment, it will degrade the DNA and the bacteria will not be able to use it. We see this exclusively in Streptococcus pneumonia, H. influenza, and Neisseria. So remember, transformation for fresh DNA. And finally, transduction refers to the process where a bacteriophage, which is a bacterial virus carrying its own DNA, will inject this DNA into the bacteria. This viral DNA will be integrated into the bacterial DNA and will start replicating the virus. If this results in a bacterial death, we call this a lytic phage, and if it didn't, we call this a lysogenic phage. If the bacteriophage did not kill the bacteria, aka a lysogenic phage, it will give the bacteria some superpower. So the bacteria now can produce exotoxins. Alright guys, that's everything for today. I tried to explain everything as much as I could. Hopefully I made this easier for you, and
This video is not well designed, so there are some typo, mistakes, inaccurate statements in it. So, but it's useful to help us understanding these topics on transformation, conjugation, and transduction. Okay, I'd like to spend a couple minutes to talk about other type of gene transfer. This is not in your textbook, but it's a recent discovery in it's a recent discovery in recent years. So this paper was published, I guess, in Nature or Nature Microbiology. They studied the type 4 pili of Vibrio cholera. when we talk about pili, we only introduce two types: the ordinary pili and the sex pili. But there are actually other types of pili. This type 4 pili is a different type. Okay, it contains several important proteins like QQ. Okay, this is a type 4 pili, which can grow from the cell wall to the environment. Then the pili is also able to retract into the cell. When this pili extends from the cell, it can actually grab molecules from the environment, then bring the molecule back into the cell. So let me show you how this work did. They use the type 4 pili. They try to label the type 4 pili with fluorescence so that they can actually observe the behavior of type 4 pili. Okay, they label the type 4 pili with green fluorescence, also the cell with green fluorescence, and you can see the behavior of the type 4 pili. It can get back and bring something into the cell. This red color is the extracellular DNA. They label the extracellular DNA molecules with red fluorescence. And you can see this cell using the type 4 pili to grab the DNA, bring it back into the cell. And you can see the accumulation of yellow fluorescence here. That's because the overlap of green fluorescence and red fluorescence. There is accumulation of DNA inside the cell. So the bacteria use type 4 pili to get extracellular DNA into the cell, a different type. And this DNA getting into the cell, they can use it as a nutrient. They degrade the DNA, metabolize the nucleic acid to get energy, or if it's homologous DNA, it's possible that it can recombinate into the genome and to express the DNA. A long time ago, when I worked in the lab, I want to make double mutation in the bacteria. One mutation is the PQ mutation. So I want to knock out the PQ gene. Then I want to knock out the other gene. So the first time I tried to knock out the PQ gene. I got it. But then I want to introduce the plasmid and to make another mutation. I couldn't get it because without the PQ gene, the type 4 pili cannot function. So the bacteria cannot get the suicide vector into the cell to knock out another gene. So I had to work in the other way. With the presence of PQ gene, I mutate another gene first, then I can knock out PQ gene. So that's my own experience a long time ago. But at that time, it was before the publication of this paper. We didn't realize the importance of type 4 pili. So important to take up DNA. Okay. After we learning this topic, we should understand several important terms, such as genetics. Genetics, we studied the heredity of some organism, and also we study the change in the organism, both heredity and the variation. And also we want to understand the term phenotype. You understand pheno? Pheno means something you can see, right? Something you can observe. So that's observable physical biochemical characteristics of an organism. And also genotype. And so first up, genome means something related to its genetic material. So that's the genetic makeup of a cell, of an organism. And there are two different type of variations. The first one is genomic mutation. So that's the actual change on the DNA sequence, which is heritable to pass it down to its daughter cells. The second type is phenotypic variation, and this is just a phenotype change without a DNA change. So this is reversible and cannot be passed down to its daughter cells.